Hello everyone, welcome back to our channel CSIR Net GATE Life Sciences. If you are preparing for GATE Life Sciences 2027, then this is one of the video which is most important video for your preparation, especially for the biochemistry. Today we are going to discuss the complete biochemistry syllabus of GATE Life Sciences 2027. Uh, this video might be slightly longer because we are going to discuss each and every component of the syllabus in detail, and then their strategy also we'll see it. What are the different topics you should cover so that you will get the maximum marks. And in my previous video, you have already seen that biochemistry is one of the important subject which can combine with any other second paper. Uh, Many of the students is asking us like which topic are going to be included in biochemistry, what are the different chapters are the most important chapters, and what should be they studied in details, and how we a beginner should start for their preparations. So in this video I'll explain each and every topic of the official syllabus. I will show you the official syllabus from the GATE website, which is given by ITeach, and I'll also tell you the which concepts are frequently asked, and also share some preparation tips. So watch this video till the end, and don't forget to subscribe if you seriously like the video. So let us begin the content. So this is the web link I have given you, and this is 2026 GATE syllabus, which was conducted by IT Guwahati. So in the biochemistry paper, uh, we have six different units, and these are the six different units. So if you see the first unit, where we are having different biomolecules. Then second units we are having enzyme kinetics as well as the metabolic biochemistry. Third, we are having different type of biotechniques, and fourth we are having cell structures, protein targeting, and transportation, cell transduction, and signaling system, you can call it. Fifth unit we are having molecular biology, like DNA replication, transcription, translation, DNA damage, and the some of the recent techniques like microarray, RDT, and PCR. And the last unit we are having immunology. So this is the syllabus which is given by the GATE, and each topics we will discuss today, which is derived from the official GATE syllabus. But for our betterment, for our better study, better planning, uh, the biochemistry syllabus I have divided into seven major units, and these are the seven major units like biomolecules, enzymology, bioengineering and metabolic biochemistry, you can call it, molecular biology, cell signaling, immunology, and biochemical techniques. So I'll cover these seven different components. If you complete these seven units thoroughly, certainly you have covered the entire biochemistry portion of the GATE. Now, one by one of all these seven different units we will discuss, and then also we will talk about the strategy to cover and the important points. If you have any questions, you can write in the comment box, and then later on we may have video on different components also. So the first unit, which is biomolecules, this is one of the most fundamental and the frequently asked unit. Roughly, questions are almost every year is going to be asked from the carbohydrates, lipids, proteins, and the nucleic acid. These are the four important biomolecules we are having. And if you see here, I have mentioned these are the different components, different subunits, you can call it, under the heading of each different four biomolecules. And then some additional things you should also read about, like if you know, if you want to see the carbohydrates, there we talk about all the different category of carbohydrate like monosaccharide, disaccharide, polysaccharides, and each components. So here where we should understand their classification, stereochemistry, then different bonding patterns, biological functions of the biomolecules. So normally in this case, questions often test the conceptual understanding of these molecules. At the same time, lipids, there normally we should study the fatty acid, triglycerides, phospholipids, glycolipids, cholesterol, and steroids. And clinical importance is very important in this case of lipids. In the case of uh, proteins from the another very high weightage topics. So here you should study the amino acids with the peptide bonds and all the four levels of protein structures like primary, secondary, tertiary, quaternary. And in this case, additional things is you should understand the process of protein folding, protein denaturation, the role of chaperones and some other protein folding components. So at the same similar time, you should also read the degradation portions like ubiquitination and those processes. And when we talk about the amino acids, their calculation of charges, and then isoelectric point calculations, deuterions, these are some other important points. Similarly, for nucleic acids, where we see understand the you should read the different types of DNA, RNA, their structures, bonding patterns, packaging of chromosomes, nucleosomes, organizations. Some of the components are because if you study here, these components are also going to help you in the molecular biology, where we will talk about the DNA replication, transcription, and translations. So this is in the first unit. Similarly, if you go to the second unit, which is according to our classification, it is enzymology. So enzymology is among the highest scoring unit, and normally focus on the enzyme classifications. So here I have mentioned all the six different classes of enzymes. As of now, we are having total seven classes. Additional class has been included later on, that we will talk later on. So enzyme classification, then enzyme kinetics, where we are having different kind of plots like mechanism and equations, Km, Vm, Vmax, Line-Weber plots, and different types of inhibitions. And in all those different type of inhibitions, how it is going to affect, what will the Km and Vmax so those portions. Also prepare the enzyme regulations. There are different type of regulations like feedback inhibitions, allosteric cooperation regulations, cooperativity, covalent modifications, isoenzymes, and their clinical importance is very important. And in this case, you should practice also numerical, and some graph based questions are very common. Then if you'll and go and see the like regulations, I have mentioned almost all the regulations. Then the next unit we are having, that is what we call it, biomolecules. In the sorry, not biomolecules, it is metabolic process or bioengineering, you can call it. uh, in this one, if you'll see, because it is one of the largest unit, you can call it, other than immunology, because you are going to study the metabolic process of all the biomolecules plus energy consumption, energy productions. Okay, so this unit is very large as well as extremely important, because it explains how living organism produces, stores, transfer, and utilizes their energy. Nearly every biological process, from muscle contraction to nerve impulse transmission to DNA replication to protein synthesis, depends on the efficient generation as well as the utilization of energy. So every year, if you see the previous year questions, Great Life Science includes several questions from this unit, and the most questions are conceptual and the pathway based, while some requires understanding regulatory mechanisms and energy calculations. Therefore, instead of memorizing pathways, focus on understanding how and why each pathway operates. So these units you can divide into five different subsections. Yeah, five different sections. The first section is bioenergetics, and you can call it thermodynamics portion also. Then we have metabolic process for the carbohydrate, lipid, amino acid, and the nucleic acids, or you can call nucleotides. So, if you see the bioenergetics, we will begin with the bioenergetics, the study of energy transformation in the biological systems, where different type of processes And important processes, some theories like electron transport chain. You should understand their components, how electrons moves and their requirements, proton motive force, chemiosmotic theory, oxidative phosphorylations. These are some of the important components. Then second portion is carbohydrate metabolic processes. So, if you need to understand how cell utilizes glucose to generate energy, then you should study the glycolysis, gluconeogenesis, glucogenesis, uh, glycogenolysis, citric acid cycle, pentose phosphate pathway, or you can call it the hexose monophosphate shunt. Uh, so these pathways are important because, and not only the component, you should also understand the energetics of these pathways, their regulations. So these are some of the important portions like energetics, uh, their substrate, their regulations, and metabolic disorders associated with these metabolic processes. Then comes to the lipid metabolic processes. In that one, we have some less pathway like fatty acid oxidations. There we have beta oxidations, alpha oxidation, gamma oxidations, different type of oxidations you should study. Then fatty acid synthesis, ketogenesis. Uh, you can call it ketone body formations. We have three types of ketone bodies that we will talk later. Cholesterol synthesis and their regulations, and lipoprotein metabolic processes. Um, we have five different type of lipoproteins like chylomicron, HDL, VLDL, LDL, IDL. So understand all the different metabolic processes for lipoproteins. Then we are having amino acid metabolic process where different kind of processes like transaminations, deaminations, uh, utilization of removal of ammonia from our body through a process called urea cycle, or it's also called as Krebs-Hanslet cycle. That is normally for detoxifications, and then normal nitrogen metabolism. And the last one is nucleotide metabolic processes, where we have purine synthesis, pyrimidine synthesis, both salvage pathway as well as the de novo synthetic pathway. And then all the disease associated, we call it inborn metabolic disorders associated with all these five metabolic components. And in each metabolic pathway, there are some regulatory steps. So you should remember those regulatory steps, regulatory enzymes, because that is important uh, for you to understand. So when you are going to prepare these units, there are several. So don't memorize them. Instead, try to understand why each pathway exists. Learn where these pathway takes place. Identify the rate limiting enzymes. Then understand the regulations by hormones as well as the allosteric effectors. Practice the drawing pathway repeatedly. You can take the printouts of the different pathway and paste it in front of you, so that whenever you will see, you can revise once. Then also revise ATP yielding from the different important pathway, and solve the previous year gate questions after each pathway. So bioenergetics and metabolic is one of the most rewarding unit in, and it is easy also because here mostly conceptual questions comes. If you clearly understand energy production, metabolic pathway and regulation, and biochemical integrations, you can confidently answer many conceptual and application based questions. So understand, visualize, revise, practice, and succeed. The next unit what we are having is the molecular biology. Uh, in the case of molecular biology, the like we have till now we have done biomolecules, metabolic processes, and enzymology. So that help us to understand the chemistry of life. And now we are going to see the molecular biology, which explains the how genetic information is stored, copied, expressed, regulated, and manipulated inside the living system, living cells. So every year, GATE Life Sciences asks several questions from this unit, and most concept questions are conceptual, but they can also be application based. especially from the recombinant DNA technology and molecular techniques, because nowadays we have lots of different techniques. So this unit can be divided into 6 different subtopics, or 7 different subtopics, you can call it, like DNA replications, DNA repair mechanism, repair system, transcription, translation, gene regulations, and the RDT, recombinant DNA technology, and lastly you can have different kind of applications. So when we are talking about the DNA replications, there are a number of enzymes. We will begin with DNA replication because that is one of the uh, first pathway for the central dogma. So the process by which cell duplicates antimony is called cell divisions. And there are so you should understand all the different kind of component associated with that one, like different kind of enzymes, different kind of proteins, different strands, how this system replication process starts, how it ends, and how it works. So after DNA replication, when we are talking about the DNA, when you talk about the DNA replication, that time also telomerase must be understand. Then when this DNA repair mechanism, so we have uh, 3 important types of DNA repair mechanism, like mismatch repair systems, nucleotide excision repair systems, and base excision repair system. And some additional system we are having like SOS repair system and homologous recombinations. So again, the process of these repair mechanism must be understood. Uh, in case of transcription, again important is RNA. There are different factors, different transcription factors, their enhancers, and the transcription modifications like capping, tailing, splicing. These are important things, and how these occurs and why these occurs. Again, energetics. Uh, Last, then we are having translation. Translation is a protein synthetic machinery. So there you should understand the genetic codes. You should understand the Voet hypothesis, because you are going to talk about genetic codes. There we are having a Voet hypothesis also. How protein synthesis starts, how elongation takes place, and how it is going to be over, how protein peptide bonds forms. So you should understand all these things. And lastly, you should understand the post-translational modification, because in case of translation, post-translational modification is one of the important steps. Fifth is gene regulations, which we also call as operand system. So there are 2 important operand system we study, that is lag operand and the trip operand. Then there are different kind of systems which regulates it, like epigenetic regulations, chromatin remodeling, and so all you should understand all those modification regulatory systems. In case of recombinant DNA technology, uh, different kind of restriction enzymes, vectors, different PCR, as well as more than 14 different types of PCR variations. Variable means a different kind of variants of PCR we are having. Different sequencing, DNA sequencing techniques, and gene editing tools like CRISPR-Cas. Nowadays we are using too much. So those tools you should understand. And after that one, we should study the application force of all these different components. So means whatever DNA replication, transcription, translation. So if you want to master this molecular biology portions, you should understand each process step by step instead of memorizing it. So don't memorize it, but try to understand it. Draw diagrams of replication, transcription, translation, and regulatory repeatedly. Compare the prokaryotic and eukaryotic systems, because many of the details explanations we are having for the prokaryotic system, we should also understand about the eukaryotic system. Memorize the important enzymes and their roles, their functions. Practice the operand based questions, because that is one of the confusing portions. Revise different techniques and solve the previous year questions. So, molecular biology is one of the highest scoring units. A strong understanding of DNA replication, repair mechanism, transcription, translation will help you to answer a large number of conceptual, because many of the time, many of the questions comes from this portion. The fifth unit what we are having is signaling mechanism, signaling system. So all this unit is little bit smaller than the molecular biology or metabolic units, but it is important in yet life sciences has increased significantly over the last few years. Questions are frequently asked from the signaling pathway, receptors, secondary messengers, apoptosis, and cell cycle regulations. So, cell signaling is a process by which cell communicate with each others. So, here I have divided this subunit into 7 different portions. Like we have different repair systems. 2 repair system only I have shown here. GPC are one of the important G protein coupled receptors, and receptor tyrosine kinase. But there are different types of category of receptors we are having, which is present on the plasma membrane or inside the cytoplasmic receptors. Then we have different type of secondary messengers like cyclic AMP, cyclic GMP, IP3 inositol 345 triphosphate, diacetylglycerol, calcium, a number of other secondary messengers. What are these secondary messengers? How these works and what are their roles? And then some other important regulatory signaling system, signaling cascade you should understand, like MAP kinase pathway, JAK STAT pathway, PI3K AKT pathways. This works for many of the regulations of our body also. Then you should understand the calcium signaling, because many of the signaling processes starts or ends or the through works through calcium molecules only. Then apoptosis. So we have both two types of apoptotic pathway, intrinsic as well as the extrinsic pathways. To understand all the pathways. In case of cell cycle, the important portion is the regulatory system, regulation of the cell cycle. That is the cyclin and CDK mediated regulations. So understand that one. And then some important key regulators, like either it will be oncogenes, tumor suppressor genes, CDK or cyclins. So once because this is one of the interesting topic, that is you can call it cell signaling, which increase your interest in the process. So if you want to be expert in the cell signaling, draw each signaling pathway repeatedly, understand receptor activation rather than the memorizing it, compare the different receptor molecules, learn about the major secondary messengers, and solve the previous year questions. And the sixth unit we are having, that is the immunology. So immunology is the branch of science that normally study how our body protects ourselves against the pathogens such as bacteria, virus, fungi, or parasites. So every day, normally our immune system encounters thousands of foreign substances, and an efficient immune response is starts, and that is essential to maintain our health. If you'll see the recent years GATE questions, that is consistently included questions from immunology, especially from the innate immune system, adaptive immune system, antibodies, MHC, major histocompatibility complex molecules, MHC1, MHC2, all the 3 types of complement pathways, and then different kind of immunological processes like ELISA, monoclonal antibody production, and vaccines productions as well as the vaccines function. So, if your concepts are very clear, these units can be highly exfoliating. So here, very small portions has been divided into 15 different subunits, subcomponents, and so innate and adaptive. That is the two branches of the major branches of our immune system. Uh, Antigens, we have different type of antigens, their properties, characteristic feature you can call it, or then antibodies, 5 different type of antibodies, their productions, then those classes, and then we have two important you can call it lymphocytes, that is the B lymphocytes and the T lymphocytes. In T we have different subclasses like cytotoxic T cells, T helper cells. We have two major histocompatibility complex, MHC1, MHC2, how their processing takes place, how MHC present those antigens, either it will be exoantigens or endoantigens onto the plasma membrane of antigen presenting cells. 3 important complement systems like classical complement system, lectin complement system, and the alternative complement system, and how ultimately it results into the membrane attacking complex formation, which is responsible for the cell lysis. as well as the importance of complement system in the opsonization, inflammation, and the cell lysis. Then four important types of hypersensitivity: type 1, type 2, type 3, type 4. Uh, then vaccines, different classes of vaccines, different types of vaccines, how it works to activate your immune system. Monoclonal antibody as and hybridoma techniques for the production of monoclonal antibodies. ELISA, different types of ELISA, different classes of ELISA, like direct, indirect, sandwich, and competitive ELISA. Other immunological techniques like flow cytometry, and some immunological disorders, because autoimmune disorders, immunodeficiency disorders, allergic disorders, grafting, transplant rejections. So you should understand all these important, because in case of immunology, uh, you may encounter lots of different types of new terms. So try to remember those terms. So in this case, again, pathway diagrams are very important. Uh, Prepare tables for immunoglobulins that will help you to compare it and memorize it. Again, uh, in complement systems, you have pathways. So memorize those pathways and revise immunological techniques like ELISA and flow cytometry and solve the previous year questions, because immunology is one of the conceptual and giving you very high scores. Rather than memorizing facts, focus on the understanding how the immune system recognizes pathogens, activates defense mechanism, eliminates antigens, infections, and the develop long-term immunity. So, and the last unit what we are having is the biochemical techniques. So, the many students underestimate this unit. It is actually one of the most scoring section of the syllabus. The good thing about this unit is the most questions are direct, application based, and the concept oriented. If you understand the working principle of different laboratory techniques, you can score excellent marks through this unit. So biochemical techniques are the tools that scientists are used to for the study DNA, RNA, proteins, enzymes, cells, and biomolecules. Whether you are working in the research laboratory, biochemical industry, hospitals, or pharmaceutical industry, these techniques are very important. So here we have some of the important techniques which have included is different types of chromatography. Uh, their process you should understand. Electrophoresis, again different types of electrophoresis. Centrifugations, again different types of centrifugations. Spectroscopy, their classifications, and some molecular techniques. I have told you PCR, 14, 50 different types of PCRs. Then blottings, we have three important blottings. DNA sequencings, microarray techniques, ELISA, and microscopy. Microscopy is again important component, important unit. So these molecular techniques helps you different places. So if you want to master this biochemical techniques, focus on the understanding of the principle of each techniques and learn what each techniques is used for. Compare similar techniques in the tables, compare them and memorize their applications. Okay. So, and once we have covered all these 7 units, so we are going to discuss now that what are the high weightage topics or high scoring topics. So, once we have completed all the entire biochemistry syllabus, the most important questions each student asks is which topics should I focus, and if I have limited time, uh, the truth is, every topic in the syllabus is important. However, based on the analysis of the previous year grid questions, certain topics appears repeatedly and carry relatively higher weightage. If you are preparing strategically, these are the topics you should prioritize. Let's them one by one, like enzyme kinetics, one of the most important. Then glycolysis, you can call it the biochemical pathway, glycolysis and TCA cycle. And then because again I'm not talking about the glycolysis, because we have already discussed that in glycolysis you should understand the intermediates, intermediate metabolites, their rate limiting enzymes in ATP consuming steps, ATP producing steps, NADH producing steps, regulations and other things. So after TCA cycle, oxidative phosphorylations, where the five different components like four different complexes in ATP synthase and protein motifores. And there are questions, sometimes questions asked from the inhibitors of these oxidative phosphorylation. Molecular biology, related topics like replication, transcription, and translations. Immunology, PCR and their variants, chromatography, electrophoresis, cell signaling, and metabolic processes. So these are the some of these topics appears repeatedly in GATE examinations. So when you prepare strategically for high weightage topics, if your exams are approaching, follow this is the steps. Like finish all high weightage topics first. Prepare very concise notes for each topics. Solve the previous year questions topic wise. Revise repeatedly. Take mock test as well as to identify the weak area. And remember, solving previous year question is one of the best way to understand the examiner's mindset. Okay. Uh, so, in this, before going to discuss about the books, so final advice is if you have enough time, study the entire syllabus because GATE can ask questions from any topics. However, if you have short time, less time, make sure these high weightage topics are absolutely strong. So, develop concept clarity, regular practices, multiple revisions, and good time management. Now, when we are talking about the important books, lots of books, because yesterday also I have talked about, don't read multiple books at a one moment of time. So, because many students are confused about the books also. So, which books should I focus? Which books should cover the GATE Excel? So, this is one of the important questions, but because many students waste valuable times to collecting too many books. So if you don't need to study every available books, whatever I have shown here also. In studying, choose one or two standard textbooks, understand the concepts thoroughly, prepare your own note, and solve previous year questions regularly. Like some of the important books which I have studied during my study, that is Lehninger, especially for DNA replication, transcription, and translation to understand. Harper's for metabolic processes, and for clinical concepts, you can study Voet Voet or Gerald Karp books are also there. Then to understand the molecular biology, Albert's molecular biology, or you can call it the cell. And then this is the James D. Watson's molecular biology of genes. For immunology, we have either Kuby or Voet you can study. Kuby is most preferred book that you can study. And for molecular techniques or analytical techniques, you can go for Wilson Walker's this principle and techniques of the biochemistry, and try to get the recent books. Sorry. Then in the last, what should be the strategy? So now, because we have reached one of the in the last portion of this video, so how to prepare effectively for GATE Excel biochemistry. Um, so first step is to finish your syllabus. Once get a complete syllabus. So many students keep revising the same topics repeatedly and never finish the syllabus. Don't make that mistake. First goal should be the get an overview of each unit from an official syllabus. Then second step is to prepare concise notes. After completing each topics, prepare your own concise notes. You can get it from some of the note providers also. Uh, then in this case, when you are preparing the concise notes, definitions are important, diagrams, pathways, formula, enzymes names, the regulatory steps, and some tricks. You can use mnemonics also for this one. Third important step is for preparation is solve previous year questions topic wise. So once you finish a topic, immediately solve the previous year questions related to that topics. Like if you are studying the enzyme kinetics, solve all previous year questions from Enzymology. After completing molecular values, solve questions from the DNA replication, transcription, and gene regulation. So this will help you to understand the examination pattern, identify frequently asked questions, concepts, learn how questions are framed. Next step is revise weekly. Revisions is the secret behind every high GATE scores. So no matter how well you are studied today, you will forget most of them if you don't revise regularly. So I recommend following a weekly revision schedule. Every week, revise your notes, revise important pathway, revise important enzyme kinetics, molecular biology concepts, and especially if possible, revise through important diagrams. Then take mock test, and the last one will be the analyze mistakes and improve your weak area. uh, so these are the different things in the last. The biochemistry section is one of the most scoring optional subject for GATE life sciences. If your fundamentals are strong and you revise regularly, scoring more than 25% marks out of the 30 in this section is an achievable target. So don't try to memorize everything. Instead, focus on the understanding concepts, pathway, mechanism, and their applications. So that's all about. So uh, thank you for your watching. If you found this video is helpful, like the video, comments which biochemistry topics you want me to explain next. Share this video to your friends who is preparing for GATE Life Sciences. And if you like the video, subscribe my channels. Thank you for watching. See you in the next video.